Okay, welcome to the Theoretical and Computational Biophysics Seminar. It's my pleasure to host today Dr. Guillaume Lamoureux from the Department of Chemistry at Rutgers University. Guillaume received his PhD under the supervision of Lamoureux at University of Montreal when he worked on polarizable force fields, if I'm not mistaken. And then he moved to Pennsylvania to work with
Michael Klein. I was with Michael Klein. Yeah. Oh, sorry, Michael.
Yeah. With Michael Klein when he worked on QM simulations. I'm so sorry. This pain is killing me. Maybe I should speak slowly.
Yeah.
Metalloenzymes and
or do you want me to?
I think I might be able to.
Okay.
And then he goes back to his home country and joins the faculty of the Department of Chemistry and Biochemistry at Concordia University before moving back to the United States, where he moved to the Department of Chemistry at Rutgers.
Mm-hmm.
So as you can tell from his trajectory, he has been working in our field very closely, force field development, simulations. But now he has turned around and sort of working on how to apply machine learning to structure prediction of proteins and protein-protein interactions. So we look forward very much to your talk, predicting protein structure using functional data.
Hey.
For coming out.
Thank you very much. And I'm sorry to put you through this.
Sorry to butcher your introduction.
Yeah, I have like shivered a little bit. I know that.
I'm sorry.
So right. So and it's been a pleasure to meet you. Yeah, I've had really interesting discussions this morning and this afternoon. It's a pleasure also to visit the institute. So yes, I've been doing a slow transition from molecular dynamics to machine learning, not with the purpose of completely, I mean, not because I don't like molecular dynamics, but because I think there's a lot that can be gained from bringing the two together. So this is the short version of the talk, of the title of the talk. I mean, so the longer title could be Predicting Protein-Protein Complex Structure. So the structure of protein-protein complexes using functional data, but specifically I will talk about interaction data, right? So, but I think it's more general than that. So I wanted to have a more, let's say, like seductive title that would kind of generalize a little bit. So I'll be presenting the work primarily of Georgi Derivianko, who was a postdoc in the lab in Montreal before I moved to Rutgers, and also of Sid, who's a graduate student in the lab. So both have been working together and kind of have pushed the work we're doing also. But other people have been involved in other aspects, which I'm not going to present. But yes, you'll see those names coming back in the slides. So for the purpose of predicting protein structure from let's say basically how do you predict the structure of a protein if the only information you start with is sequence, right? So there's a I mean conceptually we imagine that there's a path that takes us from sequence to structure. That may be difficult, but as a kind of postulate, we assume that there is a structure to discover from a sequence. We know that this is an imperfect let's say assumption, because not every sequence has a well-defined structure. So of course here I you know, imagine that, you know, if there is a structure, it would be let's say predictable. So so from a machine learning perspective, this is a problem of essentially transforming an input into an output, right, through a series of transformations that would have to be learned or discovered. Okay, so and that's the general protein structure prediction problem. So which has been a big challenge for many years. So what has started to work recently is an approach that's a bit indirect, but very successful, which is to make use, of course, of structural data that's available. So this is, of course, an important set of data to train on, but there's also a lot to learn from sequence on its own. And the logic here is that if you try to understand the language of protein sequences, and if you manage to do that, you may end up with a fairly abstract representation of protein sequence or protein sequences in general that ends up being extremely useful as an input to predict the structure. So, and the idea is that the only way to really, for instance, understand why a protein sequence is the way it is to understand that it has a structure, it has a function, it has an evolutionary history, and so on. And models have recently emerged that use precisely that idea. They're based on the analogy with language. So if you haven't seen those papers in the literature, so there's ESM Fold by a team from Facebook, mainly, who have trained a large language model to represent protein sequences, and that model generates a representation that in turn is extremely useful to predict three-dimensional structure. So they've developed both the language model called ESM-2, and then that model was used into the ESM Fold model that does structure prediction. Another one along the same lines from a team at Columbia and at Harvard, led by Mohammed Al-Quraishi, where they use again a large language model, AminoBERT, to create a representation of protein sequences, and then that is used to predict protein structure again. So these are very successful. They're not exactly how, let's say, AlphaFold or Rosetta Fold work, but they're in a way just like a variation on the same idea, but the difference is the major difference, I would say, is that AlphaFold and Rosetta Fold do not directly try to learn a representation of the sequence. They use the sequence and also a multiple sequence alignment, and this is also a very useful way to get started on the protein structure problem. Okay, but of course these are large models, and they work, I would say, in part because now you train on both structural data and sequence data in this kind of unusual way, right? So our question, what we tag onto that picture is another option here, which would be, okay, can we get better at predicting structure, for instance, if we were also trying to predict the function that predicted structure would have, right? So now here the kind of endpoint is some predicted function, which allows you to use all sorts of functional data you might have, coming from proteomics, for instance, or I mean, any high throughput assay, is actually usable here, as long as you have a good model to make a prediction about it. So the question now becomes: is this actually a helpful thing to provide to a model if you try to improve structure prediction? So we've been playing with those ideas from let's say for a while. So our kind of early attempts, not at predicting function, but at predicting, I mean, really just scoring decoys or protein structure decoys, was done in 2018. But the idea here is a bit similar. Is that so if you have a possibly correct, but maybe incorrect structure for a protein, can you tell how far it is from, let's say, the native structure, right? So by giving it a score, that would be better if you're closer to the native structure. And we've developed that model, which is that's the details of the model, but I mean, it's represented by that gray triangle here. But of course, it has lots of layers. But essentially, what you have here is a giant data funnel that boils down a three-dimensional representation of the protein. So basically, you take that structure and you project it on grids, where each grid contains a density of a certain type of atom. So you do atom typing here, and then this gets processed down to a single number, which is a score, which you then use to rank the decoys and train the model to see if eventually it gets good at scoring the different proteins and being able to tell which one is more native-like than the other. So just a bit more details on what the input for that kind of model looks like. So I mean, here I'm not showing a very interesting protein. It's just an alpha helix. But what we would feed the model as input is more or less just the location of atoms of a certain type. So amide nitrogens are located on the backbone. So you would have a trace of of densities, where those atoms are, and so on. So those are the carbonyl oxygens and so on. Then hydroxyl oxygens, you have a few of them, so on. So we have 11 atom types like that. So each of these is a three-dimensional density grid. When we have multiple channels like that. So that's our input. And then the model has to use that information and rank the structure, right?
What is the advantage of densities versus just the position?
Because we were doing everything on a grid, we have to to spread out the location of the atom. So actually, it's convoluted with a Gaussian. So it's done on grid. That's the main reason. Right. So we designed that training procedure. So training was done on structures from past CASP competitions, for which we had both the native structure and a series of decoys of various qualities. And then we tested on additional data sets. So the model would be trained to basically get the ranking of pairs of decoys right. So given two different potentially good structures for a protein, which of these is ranking better than the other one? So that's the task that the model is trained on. And so I mean, I'm skipping here the results. So when we published that, it was performing well. So we were you know, let's say in the yeah, we were basically like getting to state of the art or close enough, but then notice the date here. So so what happened between 2018 and now is AlphaFold. So this became in a way a kind of moot like point or a task that was not needed anymore, because nobody really needs to do a quality assessment of models now. They just have to make a prediction that's much better than anything else you could do. And then if that procedure also gives you an estimate of the confidence of the prediction, then you actually have all you need. And actually, the last CASP competition has just ended that particular competition where they asked people to rank models, because there was just no point, right? So I don't think this is a paper that will get cited much more in the future, except maybe for a few ideas that we had in there. But the task it was trying to perform is a bit obsolete now. But many things are a bit obsolete now, I would say. So one thing that's nice, which I show about that work, and that remains interesting, is that once you have a model that's trained to tell you how good or bad a certain structure is, you can ask, well, what makes you think that this is a good or a bad model, right? So and there is a way to interrogate the network to actually answer that question partially. Yeah. So there's this method that's called the GradCam method, which allows you to localize, to basically figure out where in the input you might have issues or not. This is a figure from the GradCam paper, where they had trained a classifier to tell what animals, I think, or objects were on the figure. So out of the model comes a prediction that yes, there is a cat in that image, but then this GradCam method can basically backpropagate that answer and see exactly what part of the input was responsible for that cat prediction, right? So so here, so according to the model, this is what let's say makes the answer being a cat, right? But if you do the same exercise for the output dog, then it would tell you that this is what makes the answer look like dog, right? So this is a nice way to diagnose the quality of your predictions, and also to troubleshoot if something goes wrong or is kind of mispredicted. You have a sense of where it went wrong, or what let's say Q it's picked from the input that was incorrect, and so on. So we did exactly the same thing on the decoy structures that we were providing the model. So what you see here is more or less the GradCam analysis color coded on the structure of the decoy, right? So so those are all the same protein, but just predicted at different with different accuracies. Last one though is the native structure. So the network has never seen any native structure, so it was trained purely on decoys, so nothing was exactly right. But just by being good at telling apart like a better versus a worse, let's say, prediction for the model, it ended up also being good at telling you that I've never seen this one, but it looks pretty optimal, right? So, and you can tell from the GradCam analysis that well, everything is more or less uniformly blue here. So as far as the network, the model is concerned, there's nothing in the input really that could be improved to make the prediction better. So this kind of came out a bit, let's say for free from training on decoys. The fact that we can also tell when a native structure is indeed the best one. But it does point to the more problematic regions, right? So that's interesting. So moving on, then we were interested in using similar ideas for protein-protein interactions. So our first attempt at the problem, the one I'll show a bit more here, was also using this notion that if we want to tell what's the best, let's say, pose or interaction pose between two proteins, we would have to train a model to tell different structural decoys apart, right? So take one pair of proteins, generate many structures, some will be better than other, and then the model should be able to tell which is which, right? So which one's good, which one is not so good. So really the same idea for protein-protein interactions. We had an idea that was going back to that was just basically, that's just a generalization of how people have been doing protein-protein docking, rigid protein-protein docking for a while now. This is I would say the kind of like foundational paper where they did rigid docking using like fast Fourier transform. If you were to predict how those two proteins would bind and how they would come together, a simple-minded way of doing that would be to imagine two different features, so spatially resolved features. So each protein has a set of two features, right? So the first one is just telling you whether you're inside the protein or not. So this is a function of space that is 1 inside the protein and 0 outside, right? And then the second feature is telling you whether you're at the surface of the protein or not. So again, 1 if you're on the surface, or like close enough to the surface, and 0 if you're not. So it's 0 inside, 0 outside, but 1 just at the surface. And then you do a very simple thing, which is to find what's the best translation and rotation of a ligand onto a receptor, so that you would minimize this very basic and crude energy function, which does essentially two things. It penalizes volume overlap, so you don't want the ligand to be on top of the receptor, and it maximizes surface overlap. So it's just doing shape complementarity, more or less. So there's no notion of electrostatics. It's not trying to match any like charges, or hydrophobicity patterns and so on. So it's just going by the shape. But it still works reasonably well. So I mean, there are better ways to do that, but the idea is interesting. So the kind of version we had of that idea was to well, introduce more features. So why only 2, right? So let's have a list of, I don't know, 64 different features or 32 different features, which would come out of a convolutional neural network. So not just built based on very simple rules like that, but just derived from a convolutional neural net that would have to be trained. And then the energy is computed more or less the same way, by just mixing, well, or calculating the correlations between those features, which on their own is not an energy, but then you can use these correlations as input to another neural net that would return something that would be interpreted like an energy, so, or a score, and then that's the score you use to decide whether this decoy is better than that decoy, right? So that's the basic idea. Now the model itself has more to it. But that's more or less the way it goes. So each protein goes through is processed basically the same way we did for the first project. So we map the protein structure on a bunch of grids broken down by atom type, and then this gets processed all the way to just a number of features here for which we take correlations, and then these correlations, we have a number of them, like 48, and then we just mix these using a fully connected network all the way down to a single number, which is our score, right? Right, so now what goes inside here has a few interesting things I'll mention. It will come back later. So these two let's say branches or these two modules of the network are actually the same. So we completely share the weights between the two. So of course, because it doesn't matter which protein we call the receptor or which one we call the ligand. So this is just the same operation twice, right? And here it is. So this is a kind of zooming in on the operation itself. So it's a series of convolutions and then ReLUs, so all the way down here. We actually lower the resolution at this point. So there's a strided convolution here, but it's not critical. So this was more like a trick to reduce the memory and the computation of the model. But what's important here, what ended up being, let's say, critical to the training of this model is that these are not regular convolutions. They're not exactly the basic convolutions people use for image analysis. So they're SE3 equivariant convolutions. So I'll explain more what that means, but one important thing that SE3 equivariant convolutions do is that you do not have to train them on rotated versions of the same image, right? Because the protein has no notion of up and down. It should be presented. I mean, it might be presented to the network in any orientation, right? This is not a concern, right? So it's different from images where most of the time there's a clear notion of up and down, because that's how people take pictures. But for molecular objects, we need this SE3 equivariance. And these convolutional layers have that equivariance, meaning if you rotate the input in a certain way, out comes an output that will be exactly the same, but rotated in exactly the same way. So it completely decouples the orientation of the input with the actual, let's say, information content of the input itself. Yeah. So in more detail, this is what I mean, right? So SE3, if you're not familiar, is a group of translations and rotations in 3D space. So if you talk just about rotations, it's the SO3 group. But SC3 also includes the translation. So regular convnets are translationally equivariant by construction. That's what a convolutional network is. But the rotationally equivariant part is an additional feature. Okay. Right, and specifically, if you have a function f, let's say a layer of like a convolutional layer, it's SE3 equivariant if the outcome of translating the input is the same as translating the output of an original input, right? So that's what I said, just in the mathematical form. The good thing here is that you will learn much more effectively any pattern you might encounter, regardless of how it's oriented in space. So any arrangement of atoms that you see once in any orientation, if you learn it, you'll have learned it in all possible orientations or at any position in space. Okay. Right. So this was actually this is a very important paper for anybody who wants to do let's say to do machine learning of molecular objects on grids. They have now I mean the same idea can also be done on graph-based data. So so there's an SE3 equivariant version of graph convolutional networks and so on. Right, so that's what is happening in those convolutional layers. Okay. Now, how do we train this? So it's just that same idea that you know, the task here is to rank structures of complexes that may be good or bad and so on. So we started from a database of high resolution complexes. So some were homodimers, some were heterodimers, and then to introduce a bit of conformational variability, we took the complexes apart and then generated decoys of the individual monomers using this this distortion method that will it's a bit like it's just normal mode, but while also, I mean, it moves the atoms along some normal modes, but also preserves all the important distances and angles. So so what you have here is just a remixed version of that original receptor, each one looking a little bit different. Then we well, we do the same for the ligand, and then we redock all of these. Just we cross dock them, right? Using an existing rigid docking algorithm. So and these are our decoys. Okay, so we take these, compare them to the known structure, and then assign them a score based on this Capri like quality classes, right? So and the task for our model now is to actually get that ranking, let's say, correct, right? So is this a good one? Is this one better than that one, and so on? Okay. Right, a big table of numbers, but here I think the idea, so it's a comparison with just one method that was state of the art at the time. So and these two groups of columns here are basic models that we've developed here. So this is the regular CNN without the SE3 equivariance, and this is with the SE3 equivariant version of the confidence. So apart from the fact that well, the performance is overall better if you use SE3 equivariant confidence. So that's you know, but there's nothing really shatteringly different compared to a regular CNN. However, the training is much easier, and also the number of learnable parameters is much smaller. And that makes sense because we don't have to learn all possible orientations of the same pattern, right? We're actually doing well compared to ClusPro. So these different like indicators of performance. So I don't want to explain in detail, but the first column here, you see the higher it is, the better. So the closer to 1,000 it is, the better. And the minimum IRMSD is also, I mean, the closer, the smaller it is, the better. Okay, so this one should be as high as possible. That one should be as low as possible. And so we're doing fine, except for antibody antigen complexes, which is a part of why we're focusing on that now. So because that's the antibody antigen. It's a prediction is a challenging problem because the interaction interface is very small. So it's hard to get that right. So ClusPro does particularly well there. So we're not sure exactly why, but it does have a special way to deal with antibody antigen pairs. So there's some of that in that increased performance here. Right. Okay. Now, so that's fine. But again, if you've used AlphaFold, you could say, well, I mean, can you do that with AlphaFold multimer and so? And yes, you can. So again, there's a bit of a like playing catch up that we need to do. So so let me talk about this now. So can we use non-structural data to improve structural prediction? Okay, and so that's the question. So sure, we can learn to predict the structure of a protein complex by training on known complexes. Okay, so that's the obvious way to do it. But do we need to train on decoy structures? So I would say the procedure, the task here is a bit artificial because we have to generate all those bad structures just to ask the model which one is better and so on. So it's computationally expensive and also not quite let's say I mean it doesn't look like it's the best way to do it. Why not just learn from the structures we actually see, right? Why kind of imagine all those bad structures just to quiz the model on you know, and create some kind of multiple choice, you know, where it has to find the right solution. So so another idea which I'll explore in the last in the next I don't know 15 minutes maybe. So can we train a structure prediction model just on the knowledge that two proteins interact? Okay, so it's a bit minimalist, but you know, if you show the model two protein structures of monomers and tell it, oh, these two actually form a complex and these two don't. And if you repeat that for lots of complexes, is that enough information to actually train a model to generate the correct structure of the complex, right? The answer we were imagining is yes, because the only way to answer that question reliably is to have some kind of mental image of what happens to those two proteins, right, when they come together. So we didn't want to answer that question on the full data sets of proteins. So we kind of went first with a toy version of the task. And this was an excellent idea in the end because a lot of things had to be clarified, right? So we created that two-dimensional world. So proteins there are just two-dimensional shapes, literally the shapes you see here. And so we just posited an energy function that is very much like the one I was like showing before. So maximizing the interface overlap and minimizing the shape overlap, right? So and then with that energy function, you can then predict which two shapes are going to come together and how exactly, right? So so with those shapes generated algorithmically, really, so we generated two slightly different data sets. One where you know the pair of proteins or shapes is interacting, and the question is how exactly are they interacting? So what's the interaction pose? So I'm calling that doc2dip interaction pose. And then there's a second data set where you actually don't know whether the proteins interact or not. Okay, so the input would be those two shapes, but then the piece of information you're provided is 1 if they interact and 0 if they don't. And here that threshold is determined by computing the equivalent of the free energy of binding and just setting a cutoff. So if you're the cutoff here is a minus 100%. So if you're below 100, minus 100, not 100%, minus 100. If you're below 100, you call that an interaction, a positive interaction. If you're above, you're always, any two shapes will be, it's like sticking, at least to some extent. So, but if you're above that cutoff, then you're not interacting. Okay, so So those are the data sets. So the question now is, can we use that data set to learn the task that we could answer directly from this data set, right? So the idea is that we have a lot more data, in real life that are coming from high throughput assays of binding assays, where all is just whether an interaction was detected or not, right? Yeah.
How do you come up with that -100?
Oh, just to it was very arbitrary because there's a complete, there's a complete gray zone. So we just wanted a certain fraction of positive interactions versus negative interactions. Yeah. So it's anywhere you put it. So I guess it mimics more or less the experimental sensitivity that you would have when you think you've seen an interaction. Right. So in practice, right? So it's never so clear what's happening because any two shapes, right, could interact in different poses that are local minima of the energy function, right? So, but presumably one is better, and if it's good enough, then its free energy would be below that -100 cutoff, and then we call this a positive interaction. But there's always going to be, suboptimal interactions, like you see here. So now the correct docking pose according to that energy function would be here, right? And the correct, the binding free energy would be that number here, which is below the energy minimum because there's some kind of entropic contribution also. That's being added to that. So the model we use to train on both tasks actually is very similar because the idea is to see if we can kind of reconcile those two artificial data sets, and if both of these data sets would lead us to learn the same underlying energy function. Okay, so here we're in 2D. So those convolutional neural nets are equivariant in 2D. So in goes the shape of each protein, right? Each shape here. This gets encoded into just only two representations. So we have only two features here. And then we perform the same correlation operation that we did for the 3D docking. So this star here represents let's say taking the correlations of every feature with every other feature from the other protein, right? So one additional operation we do here is that just to process all possible poses at once, we take those features and we rotate them by increments of 1 degree. So over the entire circle, right, we'll have 360 pairs of features, each one being a grid of 50 by 50. Okay, so it's a pretty large tensor, and this tensor will get bigger in 3D. But the net result here is that you end up having calculated the energy score of all possible poses of this ligand around that receptor. So for every possible rotation of the ligand and every possible translation in 2D, which is why now you have a grid of 360 by 100 by 100. So this is a big table of scores, right? But what's nice is that what this is really is just the energy. We interpret it as just the energy of docking those two shapes in any possible conformation, and then from that, using just basic stat mech, we can get to a probability, because we just take the Boltzmann weight of each energy and write as a probability, and that leads us directly to make a prediction about which pose which of these poses is optimal, and that's our prediction for the pose. And to get the free energy, we compute the partition function and take minus the log of the partition function, and that's our delta F. Okay, and once we have delta F, this is a delta F of binding of those two shapes. We can convert that to a probability of binding, and then that's our prediction whether those two shapes bind or not, right? This prediction we can train on the fact of interaction data, those zeros and those ones that we had. Okay, so we can go both ways, right? We can even joint train those two, have a model that predicts both and make sure that everything is consistent. Okay, so yeah, these are just the predictions I always mentioned. Right, so the equivariant, maybe I'll show the slide, but I think I've explained it well enough. But the idea of an equivariance, the power of an equivariant CNN is that it will, by construction, generalize any pattern learned in one orientation into any other orientation. So if for a regular CNN, each of these A letters is a different pattern that needs to be learned. So the model has to learn a separate, let's say, kernel for each of these, right? Or filter. So but an equivariant CNN would see all these different letters as just the same and like just the same thing, right, over and over. So only that pattern needs to be learned. So that's where the economy in parameter is coming from, let's say, and the ease of training. Right. Okay. So back to the 2D docking. So if we train on pose data only, and here I would say focus just on the first block here. It's not really critical that we go through all the numbers here, but the general conclusion is that it works as long as you don't have a ridiculously low number of training examples, right? So if you have like 100 examples or so, right? I mean, the energy function is fairly straightforward to learn because it's very simple here. It's really just a toy energy function, but what you see here is performance on the validation and testing set, described as a pixel RMSD. So the predictions are on average 1 pixel away from the true pose, okay, which is not much really. So you're pretty much visually the predictions look identical to the true binding pose, right? I mean, unless you use like so few examples that really you just haven't seen much of the world yet, but this is only 10 examples, right? So and then of course it degrades. Now the simplified model is interesting because it's a model where we make the task much easier by just asking to predict the translation of the pose. So we basically feed, we send that we already arrange the input in the right orientation. So all it needs to learn is how to slide the two shapes without rotating them. Okay, so we never ask for a prediction of the rotation, right? So it's a simplified task, but somehow it's enough for the model that comes out. So a model that trains on that simplified task is actually as good at solving the full task, including predicting the rotation. Even though it has never been asked that question, it has never been asked to do that. Okay, so that goes to it kind of says that there is such a strong connection between getting the translation right and getting the rotation right, that if you know how to do that one task of just translation, you pretty much know how the rotation is going to work, right? So, and this also comes, I think, because we're using SE2 equivariant networks, because these are naturally aware of how rotations work, right? And yeah, as soon as we put that model into let's say to the task of predicting the whole thing, it does it like flawlessly. It's just as good and maybe even better. Okay. Better because it's much easier to train. By the way, this trains like really fast because there's not much you're trying to predict here. So that's interesting, and this idea is actually completely applicable to 3D. Where it's much more of an advantage actually, because now you're looking at 3D rotations, right? So now let's look at the flip side of that. So what if we train the model on interaction data only? Okay, so just to predict whether two shapes interact or not. So this is a classification problem. Actually, you have to basically like spit out a 0 or a 1, right? So here the performance metrics are written in terms of accuracy, precision, recall, and the Matthew correlation coefficient. So the higher the better, right? The closer to 1 the better. So and we see again the same kind of transferability of results, which is you know, you don't have to train on the full task to actually learn as well. I mean the energy function as well. So that this group of numbers here is an interesting experiment where we basically transfer the features that were learned from the previous task where the model was predicting the pose. And I mean, if those features were good enough to predict the pose, maybe they're almost good enough to predict the affinity, right? And they are actually. The only thing we have to do is just train the last few layers of the model that were just outputting a delta F, right? So there was a total of 4 additional parameters that needed to be trained to get that right. And so all you have to do to make that model work, as well as like a model like fully trained on interaction data, is to teach it what to do with those 5 parameters, and then the performance is pretty much identical. Okay, so it's not a surprise, because we're really trying to learn the same underlying energy functions, right? But it's nice to see that it's that easy. Okay. It won't be that easy for real proteins. Now, the last experiment is an attempt to kind of emulate let's say the challenge of not having a good way to explicitly compute the free energy. So here we can explicitly compute the free energy, because our conformational space is fairly limited. It's only 6 degrees, not even 6, like 1, 2, 3. It's like 3 degrees of freedom, right? So, but if we compute that partition function using just a subset of all conformations, right? So what we get as a prediction is a very noisy, poorly converged estimate of delta F, right? Which we then try to make look as to match the real value. But somehow this is, even though it's noisy, it's enough of a signal that the model can learn the energy function the same way. Okay, so even though at no point was the free energy estimate really converged in that process. Okay, so that's also interesting. The training is much longer though. So that's just a lot of noise. Okay, I don't want to go over time. So right, okay. So I think I'll skip that. But that's just an interesting, that's a conceptual point which you might have detected already, which is if once you have a way to get once you have a task that generates a useful representation, you might want to consider whether that representation you've learned is useful for other tasks. Okay, so and this was exactly the case here. So we had we had the task of predicting pose versus the task of predicting affinity that were really, let's say, just the two sides of the same coin, right? And then, so if you train on one, you will most likely have interesting representations that will be very easy to use for another task. Okay, so that's a general idea here. Okay, it works in both directions, right? So training on pose data is helpful if you want to train, if you want to learn something about interaction or affinity, and training on affinity data is useful if you want to learn about structure of the complex. Okay, all right. So the features learned, not surprisingly, end up looking very similar once you're orthonormalize them. Okay, so these are just different maps of the two features we've learned using different tasks, and because they all end up working, for the other task, no surprise that the features are extremely similar, right? Okay, so I'll finish. We've gone back to the real protein docking problem, okay, but now with a new view of it, which actually is much more straightforward, because we see now that it's not so much about the number of features you have, it's more about let's say how you train for them, and what actually they're let's say how actually they're used to generate those predictions. So we have this kind of two-headed idea where once you have an energy function, so function of some internal coordinates that describe the complexation of those two shapes, you can really go either the structural way or the thermodynamic way, right? And both are useful to get you a better picture of that energy. Okay, right. So that's just again what the energy function looks like if you care about the details a bit more. But because it's the same for both tasks, you can definitely learn it better if you train on both tasks jointly, for instance. Okay, so that's the idea. Okay, so I'll finish by just like explaining where we want to go with this. This is more long-term stuff. Yes, proteins are interesting, and there's a lot to do about proteins, to understand dynamics and conformational variability, and post-translational modifications and salt effects, pH effects, and all the good stuff, right? And all sorts of context-dependent aspects that really are needed if you want to understand what drives an interaction, but we shouldn't forget about nucleic acids and small molecules and so on. So generalizing those ideas to any biomolecule is interesting. Scaling up, so to larger systems, and possibly along the way make, or let's say like change how those molecules and those large assemblies are represented, probably going more coarse-grained. But coarse-grained, more with a multi-scale like perspective, where you would still keep some of the important details at the atomic scale if they end up being important at those larger scales and so on. So this is a tough problem, right? So how much of the atomic scale do you keep when you go all the way up to the level of a cell, right? And we know that we do have to keep some of these details, right? They matter. And then last thing I'll mention is that there's so much data to be exploited, right? So of course, all that functional data, that experimental data is nice, but just even the structural data, the PDB itself has so much more knowledge in it than just the protein folds that we see, right? So there's a lot of exquisite details that are present there that can be learned and generalized from. So really a lot to do there. Okay, all right. I'm done. So thank you.
you very much. So, any questions? I can start. How do you make protein decoys if you have single protein? For this complex that you showed us, we move them around and we know.
So in the first way we did it was we would generate decoys of the individual proteins and then redock those decoys.
Right, but how do you make individual protein decoys? For individual proteins,
sure.
how to make a decoy?
Well,
So
so
change the structure, right?
right. How do you change the structure? Well, the expensive way would be to run MD on each one and then keep snapshots on there just to get a sense of the
Yeah,
the
but you're
the
not
structural variation. No, exactly. So we do something simpler, it's more efficient, which is that NOLV method. So that introduces random deformations similar to what you would see in an MD, but it's just yeah, it's just a continuous description of that transformation, right? And then you get all those different variations.
Yeah. Really nice talk.
Thank you.
At one point you mentioned that you have a cutoff of like 100, maybe 100 and something binding or not binding.
Right.
So in dilute experiments, when you have two protein binding, you might say that that's a factor vector, but it's in the cell, it's actually a very crowded environment. Do you think that changing that could possibly capture more crowded or student volume effects for binding?
Right. So well, that minus 100 cutoff had no units. Well, I mean, it had units, but this was all in this kind of pixelated world, right, in 2D. So so the way, so basically the assumption is that as soon as the two shapes are put in interaction, well, once you compare those, let's see if I can say it clearly. Once you put those two shapes in interaction, you compute the free energy just as minus the log of the partition function of those two shapes being so basically one shape being in the box of the other, okay, in some orientation. Yeah, so it takes care of excluded volume like that, but it does assume a standard concentration. You're right, because it's basically whatever that concentration that box corresponds to. But because it's a free energy of binding, I think we're still okay. So it's just making that. So I think it would be a bit biased if we had proteins of wildly different sizes in that box, always with the same size, because then you would be looking at right, like you know, the so-called bound state would be unrealistic for very small proteins, for instance, because you would still consider those proteins to be bound even if they're in the opposite corners of the box. You see what I mean? So, but here all the proteins have more or less the same shape, so I think it's not too bad an approximation. Yeah. So, but the correct way to do it would be to have a feature, or to basically have not only correlation, like pairwise correlations between features, but I think you would also need to have a feature that's just going to tell you how big each protein is. Or at least that can be trained that way. So then I think that would help compensate for possibly the variation in size. Some free energy calculation stuff. Yeah, but you're right, that there are considerations there.
So I have one more question. Do you think actually when you go to 3D, so this is going to be huge if you have.
So right, I kind of, I mean, I don't have hard results for 3D, but I've seen preliminary results. It's just not ready to put, but actually, yes, in 3D we run into memory issues.
I see.
So it's actually why I like this idea of training on a simplified task, because then we don't have to scan the entire SO3 grid. which can
I see.
be
But
like
you
thousands of
think actually
points.
if you
Right?
exclude the rotation, we can still capture the.
Seems so. So at least it puts us like close enough to the correct answer. Then the features we've learned, we can then move to the following task, just like predicting delta G's.
Yeah.
And we're already in the kind of right area.
I see. I see.
So yeah, but the training is so much faster.
Yeah.
If you don't try to
course.
predict rotation. Yeah, yeah.
Anything on Zoom?
Yeah, that is a question, but she can ask herself if she wants to. We can hear her.
If the sound is on, yeah.
Yeah.
I'm not sure. I'll look at the camera, I guess.
Yeah. Yeah, hi,
You
thank
can
you for the
talk,
nice
Sam.
talk. Can you hear me?
Yes, Go
Yeah,
ahead.
Yeah, so, you mentioned already, I think in your second or third slide, you go sequence to structure, and then how do you get function? But to my knowledge, there's really no good database of functions.
So function, right? So
Yeah, so
are-
my question was, like people have tried like these EC numbers. That does the classification is not fine enough. And you're right, in the PDB, it has the structure, but it also has often the ligand that it's supposed to be binding to
Right,
it.
right,
But,
right.
doesn't one almost have to sort of to create a database that has a function, the features that you would need? You can design an enzyme to do this function, like to break down like glucose, if you're saying in the main glycolysis pathway, or convert a nucleoside eventually into a nucleotide. But, I just don't know any place where we've applied AlphaFold to everything that's unknown in the minimal
Yeah.
cell, has an unknown function, and it brings us a little closer, but I find that database is missing, or or do you have an idea about one?
They absolutely are missing, and also functions is a much more heterogeneous bag.
Mhm.
So far we've used databases of binding affinities. There are some
Yeah.
decent databases for that online. So I guess I call that functions, just binds to, I guess, is a function. But for enzymatic function, it's very hard. It's usually like bits and pieces of information for certain classes of enzymes scattered all over the literature. I don't know of a good repository for these. Yeah. But the hope there is that most of the training will be let's say done using sequence and structure data. But then function is just what you need on top to kind of refine the prediction. So possibly not needing as many data points, because the models are going to be much simpler. And then what you would generate is a series of specialized models that are good at predicting this type of function or that type of function. Yeah.
Okay.
So, but there's no good. I mean, I'm just coming with let's say like ontologies for functions is tricky. So let alone populating these with data.
Okay, thank you.
So what's happening here? You go back to sequence?
Right. So I didn't fully explain, but this is a like it could be a like autoencoder type of task. So right. So either yeah, exactly. Let's say predicting the same sequence out, or maybe a task where you corrupt the data and ask for a prediction of the corrupted part. And that's usually how those representations are. Yeah.
Any other questions? Yeah. Not, let's thank you one more time. Hey.